Very quickly, we're going to finish our discussion on fatty acid biosynthesis, and in particular, we left off last time around unsaturated fatty acid biosynthesis. And as you can see from the slide, this is what we talked about last time, where in animals or in a lot of different eukaryotic cells, we are able to produce unsaturated fatty acids. Now, keep in mind that here, the unsaturated fatty acids that we're making are always cis, okay? It's always a cis alkene. We don't make trans alkenes, okay? Trans alkenes are non-natural fats, okay? They're usually produced through dehydrogenation of a saturated fat, okay? So again, natural lipids are usually cis fats, okay? Cis fatty acids. And what you should notice is that on the cis fatty acids, okay, this alkene, essentially what you did was, initially it was an alkane, right? It was a saturated chain. And out of nowhere, you put an alkene on it. Okay, so again, we discussed that it's not very easy to do chemistry on just hydrocarbons, okay? So as a result, you'll have to use some specialized enzymes. And in this particular case, these things are called fatty acyl-CoA desaturase. And what they use is molecular oxygen as the oxidant, okay? So here, you're not like fatty acid beta oxidation, where the alkene is introduced because it's alpha beta, okay? Here, it's like way, way in the chain, okay? So the mechanisms will not be the same, right? But we talked about that last time. So this is where we stopped last time. Again, this is just a chart that shows you, okay, what are the unsaturated fatty acids that are occurring inside of our body? In particular, here we're looking at mammals, okay? And what you should understand is that mammalian cells, we are able to do the delta 5 and delta 6 desaturation, and they occur in the endoplasmic reticulum. This is not particularly important for my purpose, but I just want you guys to know that this exists, okay? It exists in mammalian cells. However, what you notice is that anything beyond delta 9, like delta 12, delta 15, they occur primarily in the plants, okay? So we don't make those, right? And delta 12, okay, so for example, if you look at this guy, this is 18:2C, meaning it has 18 carbons, and 2C, meaning it has 2 alkene units that are cis conformation, and it's at delta 9 and 12. Okay, so it's on the carbon 9 and 10, and then 12 and 13. If you look at 12, okay, this is pretty much the 6th carbon from the back. Okay, so counting from the back, it's 6 carbons. So this guy is omega 6, whereas this guy is 15, and it's the 3rd carbon counting from the back. So this guy will be omega 3. Okay, so your omega 6 and omega 3 oil are typically produced by plants, okay? And of course, your fishes in the sea, they'll eat these things, and then they also have a large content of their omega 3 lipids, okay? Which is why we eat fish oil, okay? We need to take in fish oil, or we don't need to, okay? But people do that because fish oil has a high omega 3 lipid content, okay? Of course, nowadays, you guys know that they make eggs that have omega 3 by feeding chickens with omega 3 diets. 有没有看过？台湾有没有卖那种东西？就是你有买那种鸡蛋，然后它上面是标说什么这个鸡蛋里面有 omega 3的量，很少听过，是不是？像在美国那 supermarket 啊，就是你可以去买蛋嘛，然后蛋有分很多种。那最基本的蛋的分类是非常大小 ，OK， 有什么 medium size、large size、extra large size 跟那个什么 jumbo，OK， 这是超大型的蛋。然后蛋一般就是白蛋跟那个红蛋嘛，就是有颜色的那个蛋 ，OK。那后来他们就开始推那种就是蛋有不同 level。那通常在国外那种 on sale 的蛋有没有？ 促销的蛋，都是那种最便宜的，可能什么99 cent 可以买1盒啊，那种都很便宜。那贵一点的，就是有那种4块钱1盒，然后就是那种看起来就很高级的蛋，然后它上面就有说什么它里面含 omega 3的含量很高啊什么的。OK， 那它鸡蛋里面哪来那么高的 omega 3？是透过他们给它吃的那个鸡的饲料里面的 omega 3含量比较高。OK， 不是说那个抽一点 omega 3然后在里面打，不是这样，是靠那个鸡的这个自己的metabolism， 然后去吃它这样。那你要想说那样子对你的健康是真的有帮助吗 ？Maybe， OK maybe。那 depending on 你要怎么去吃那个蛋 ，OK。如果你是去 fry 那个 egg 的话，那你可能就会破坏掉它的lipids。But you know regardless， 生吃吗？对，所以就是you know people do weird things。那你们很多就是念生科嘛，那这些东西其实都跟biotechnology 有关系啦，所以你可以想说各式各样的 market out there。那很多人现在，尤其台湾还算是一个比较 developed country 嘛，所以我们平均一般人的消费能力颇高啦 ，OK， 所以你还是可以去想一些那种很奇特的 business 哦，就是说可能有些人会为了一些什么，好像有什么健康效果啦，然后买一些很奇特的 product，OK， 但是一方面也是我们国人很大的一个问题啦，就是你们常听到说那个台湾是大概全世界洗肾的那个 user 量最大的，就是因为我们很多那种Chinese 妈妈们喜欢买一些很奇怪的保健食品 ，OK， 那尤其是什么他家小孩就是长不高啊，然后就给他吃一大堆很奇怪的东西，但是那是我们很 special 的 culture 啊 ，OK。Alright, but anyways, coming back to our lecture. So again, your plants are able to do these omega-6, omega-3 oil. However, when we need them, since we don't produce them ourselves, so we will take in these lipids from our diet, okay? And then once we do that, we will be able to assimilate these lipids into our cell, okay, into our metabolism. And what is really important for us, for example, arachidonic acid, okay, so this C20 molecule, okay, so we go from C18, this guy's 18, and we come into our cell and we can do delta 6, right? So we can do that, okay? We can do delta 9, delta 6, delta 5, okay? And then now we can then elongate, okay, this guy's 18, we can elongate it to 20, okay? This elongation mechanism is actually through a direct malonyl-CoA addition, okay? It doesn't have to go through fatty acid synthase or anything like that. It's a specialized enzyme that just add 2 more carbons, okay? Once you get to C20, and then you will generate this delta 8, 11, 14. Well, you notice how it went from 6 to 8, 9 to 11, 12 to 14, because you added 2 more carbon at the front, okay, not at the end. All right, so that's why the carbons shifted. And then you can do delta 5, okay, and then you put a delta 5 alkene on it. Now you have 4 alkene units on this organic acid, and this thing is called the arachidonic acid, okay? Now, arachidonic acid is actually very important because they're used to make this class of molecule down here that's called the eicosanoids, okay? Eicosa means 20, okay? Eicosa means 20, okay? So it means that these type of molecules have 20 carbons, okay? So that's why they're called the eicosanoids. So one very good, well actually this is the metabolism to produce different eicosanoids, but you don't have to really remember a lot of different eicosanoids, but I just want you to know this word and their general shape, okay? So you start with arachidonic acid or arachidonate, this guy, okay? And what you're going to do is you can use several different oxygenases, okay? For example, here you have a cyclooxygenase, COX. Have any of you heard of this enzyme before? COX, like COX-1, COX-2. Okay, we'll talk a little bit about that, right? And it's COX, C-O-X, because it stands for cyclooxygenase. Where's the X? O-X, okay? Oxygenase. So cyclooxygenase, okay? Now, using this enzyme, you can then create this type of molecule. And then, this PGG2 can then be reacted to a bunch of other PG blah, okay, the PG whatever, okay, prostaglandin type of molecule. And these molecules typically, have a wide functions in immuno responses and a lot of different physiological responses. Okay, so these things are signaling molecules that happen in your body. Okay, so a couple of just keywords that you should remember: prostaglandin, thromboxane, leukotrienes, these type of molecules. You should know that they are participating in a lot of immuno responses and immuno reactions. So if later on you guys go take a class in immunology or physiology, you will probably talk about these more in detail. But for now, for biochemistry class, my goal is just for you to first hear these words and sort of have an idea. It's like, okay, so these things are all called eicosanoids, and they are related to immuno responses. They're related to physiological uses. Okay, that's my purpose. Okay, you don't necessarily have to know, for example, what's the difference between PGG2 and PGE2. It doesn't matter. Okay, so eicosanoids. Right now, all right. So some of the metabolic functions of eicosanoids, so all these different molecules that we saw on the previous slide, as you can see, you can have a wide function related to these different eicosanoids. For example, promotion of sleep, muscle contraction, chemotaxis, that kind of stuff. Okay, so they're related in a lot of different functions that are important for mammalian physiology. Okay. Now, you need to understand that these type of molecules are synthesized from where. Okay, so that's really my goal. All right. Since we're talking about prostaglandin, one of the enzymes that are really, really important is COX. Okay, so we saw that the cyclooxygenase is responsible for making prostaglandins. Okay, so again, what exactly are these prostaglandins? Okay. So it turns out prostaglandins are responsible for your inflammation. Okay, some prostaglandins are responsible for your inflammation. So we know that if you have a inflammation occur, okay, so inflammation means that you have cells attacking a particular region of your body. This could be due to variety of different causes. Okay, it could be contact with a chemical, it could due to allergy, it could due to viral infection, bacterial infection. Okay, so your body responds to foreign objects through inflammation. Okay, and we know that inflammation could be bad because sometimes a virus doesn't kill you, but the inflammation kills you. Okay, so it's your body's response. This inflammation is caused in part by your production of prostaglandin. Okay, so then it is natural that we can potentially come up with drugs that inhibit your prostaglandin synthesis. Okay, now you have two different COX enzymes in your cell or in your body. They're called COX-1 and COX-2. Okay, now COX-1 is always expressed, and it's expressed in most tissues, and it is responsible for physiological production of prostaglandin. Okay, so the normal amount. Now, when your cells are invaded by chemicals or viruses, foreign objects, okay, so your cells are going to trigger the production of different cytokines and endotoxins, right? So these things. In the inflamed cells, okay, then your cell will turn on the expression of COX-2, okay? COX-2 is really, normally it's not doing much, okay, even though people try to always inhibit it, and it caused elevated risk to heart attack, heart diseases. So currently it's not a good idea to always keep your COX-2 enzyme low, but we know that when you're undergoing inflammation, your COX-2 level is going to go up. Okay, so this elevated level of COX-2 will produce a bunch of your prostaglandins during inflammation. So we know that that's not favorable because inflammation causes pain. Okay, for example, when you have a fever, usually that's related to inflammation. Okay, so a lot of times we will design drugs that sort of soothe or ease your inflammation response. Okay, and a very good example, this is aspirin. Okay, it's called salicylic acid. Okay, aspirin, and aspirin is a drug that will sort of inhibit COX-2 or COX enzymes in general. The way it works is very simple, where you have COX enzyme, and inside it, it has a active site that has serine. Okay, and what you can do is you take aspirin, you throw it into your COX enzyme, and then you will acetylate. So you transfer this acetyl group onto the serine, and you form this acetylated COX. Okay, and as a result, the active site is blocked, so then it cannot make you more prostaglandin for inflammation. Okay, so this is a way that aspirin works for inhibiting COX enzymes. Okay, so just a little story, but I'm sure when you get to more advanced classes, they'll talk more about that. Right? So now we talked about the synthesis of saturated fatty acids, unsaturated fatty acids, and I showed you unsaturated fatty acids can be used to synthesize eicosanoids. Okay, and eicosanoids are important for physiological controls and for your immune system. Right? So now we're going to spend a little bit of time talking about fatty acid synthesis and their regulation. Okay, as always, regulation is quite important. So again, recall that the rate-limiting enzyme for fatty acid biosynthesis is the ACCase, acetyl-CoA carboxylase. Okay, and turns out your acetyl-CoA carboxylase can be controlled by two mechanisms. One is allosteric inhibition and activation, and another is through covalent modification, which will remember we talked about enzyme regulation like last semester or very early this semester, that enzymes can be regulated two ways: allosteric, okay, or covalent modification. Allosteric, we typically say that it's a reversible process, okay, because you have a concentration dependence. Okay, so it's reversible. It's very quick, very reversible. And covalent modification is typically irreversible because once you covalently modify something, unless you have another enzyme that unmodify it, okay, it's going to stay inactive. Okay, so this enzyme is the most important enzyme for fatty acid biosynthesis. Therefore, it's going to be the most heavily regulated enzyme. So turns out phosphorylation by AMP activated protein kinase, okay, AMPK, or cyclic AMP dependent protein kinase. Right? So protein kinases are going to be able to inactivate ACC through phosphorylation. Okay, so these guys can inhibit them through phosphorylation. So these guys, so the AMPK and the pKa, these are covalent modification. Okay, because they are protein kinases, so they can phosphorylate your ACC and cause your ACC to shut down. If we look at citrate and long-chain fatty acids, so allosteric effectors. Citrate, 为什么你觉得 citrate 可以去 activate， 或为什么 citrate 会 activate ACCase？ 如果你的 cell 里面 citrate 很多的话 ，usually 代表什么？哈 ？acetyl-CoA 特别多，没错 ，OK。那 acetyl-CoA 特别多 ，usually 也代表什么？就是什么事情？就是如果为什么什么样子的情况下你会 acetyl-CoA 很多，然后 citrate 很多这样？如果我们说 citrate 很多的话，那你的 TCA cycle 是跑很快还是跑很慢 ？Probably 跑很慢，对不对？因为你 TCA cycle 的活性可能没有很高 ，That's why 你会 accumulate citrate， 对不对？因为你 TCA cycle 如果很快速的在跑的话，那你 citrate 会不断的被吃掉 ，right？ 那TCA cycle 如果跑很慢 ，usually 代表什么事情？我们就想说 TCA cycle 它的 net input 是 acetyl-CoA， net output 是什么 ？CO2 and CO2 是 waste 啊，我不是很 care CO2。它的 TCA cycle 的 net output 是什么？NADH 跟 FADH 还有 ATP 嘛，对不对？那要是你说 TCA cycle 跑很慢，那通常都是因为你可能 NADH 已经过多了 ，right？NADH 很多 ，FADH2 很多，然后 ATP 也很多，所以 as a result 你就不会想要做更多的 TCA cycle。Therefore 你 accumulate citrate，therefore 你 accumulate acetyl-CoA。OK？ 那NADH 如果过多，那通常代表很简单的一件事情，叫做你的 cell 的 energy 很多，对不对？你 cell 的 energy 如果很多的时候，那你会想要储存，那怎么储存 ？fatty acid，OK？ 所以这还算 logical，alright？ So turns out a lot of citrate would probably indicate that your TCA cycle is not very fast, okay? Which probably means that you have already a lot of NADH that you're not using.So it indicates that the cellular state in terms of energy is pretty high. Okay? As a result, you don't want to do more TCA cycle, so you have increased citrate. Now, your ACC is going to be responsive to citrate. So citrate is going to tell your ACC saying, hey, yo, I have too much of energy, so why don't you start to convert acetyl-CoA into malonyl-CoA? Okay? And then when you get malonyl-CoA, you can make fatty acids. So it's a way for the cell to directly tell itself to start making more lipids rather than burning it. Okay? Now, long-chain fatty acids, why would it inhibit? It's pretty much the product of fatty acid biosynthesis, right? So in a way, it's kind of like a product feedback inhibition, okay? Meaning I'm making way too much fatty acid. So fatty acid is going to tell ACCs to slow down. I don't need so much fatty acid. Okay? Now, you might be thinking, wait a minute, is it possible that I have both a lot of citrate and a lot of fatty acids at the same time? That's possible, okay? That's possible. Right? But remember, one is a activator activator, and the other one is an inhibitor. So they will just sort of cancel each other out, okay, depending on who has a higher effect for the activity of ACCAs. Okay, so it is possible. Okay, so basically this is the regulation of fatty acid biosynthesis. And the last thing here is that insulin. Okay, remember insulin? Insulin is like your not your best friend, but it's again one of the very first product of biotechnology. So you guys should be quite familiar with insulin. So insulin is a molecule that tells your blood, or that tells you to start lowering the blood glucose, okay, to store the glucose or to utilize the glucose, okay? So let's see what happens for when your cell have a lot of insulin, and let's see what happens to fatty acid biosynthesis, okay? So it turns out insulin is going to stimulate fatty acid synthesis. That makes sense, okay? Because typically when you have too much sugar in your blood, your cell wants to store it, and you can store it in fat, okay? So it's going to stimulate fatty acid biosynthesis by increasing glucose uptake. That's the first thing. If you have insulin, your cell is going to start grabbing a bunch of glucose from the extracellular space into your intracellular space, okay? And then we're going to increase the flux through pyruvate dehydrogenase to produce acetyl-CoA, okay? So insulin can also help to increase the activity of your PDH. And then here, insulin will also help to increase the activity of citrate lyase, which then also, again, gives you more acetyl-CoA. Okay, so insulin promotes the synthesis of acetyl-CoA, okay, and acetyl-CoA can then be converted to malonyl-CoA, which then goes to lipids. So fatty acid biosynthesis, you have these three little things, right? Covalent modification, you have the citrate and long-chain fatty acid activation and inhibition. And then you have insulin, which can help to increase the acetyl-CoA flux. So this is the last slide for fatty acid biosynthesis. So, I'm done after this. Right? So The very last thing that I want to talk about is the synthesis of triacylglycerol. Okay, because lipids. We now talked about how to make fatty acids. Now we need to talk about how to make them into TAG. Okay, and recall that glycerol, part of the TAG, is really made from dihydroxyacetone phosphate, okay, or from glycerol if you have glycerol intake, okay. So then, essentially, you can imagine DHAP is a 3-carbon molecule, very similar to glycerol, okay, and all you got to do is put on your acyl groups, okay. So let's take a look. In order to synthesize your TAG, you need to have this glycerol 3-phosphate, okay. So glycerol 3-phosphate, and glycerol 3-phosphate can come from 2 different places. One is coming from DHAP, and obviously DHAP is a metabolite in glycolysis, okay, so that's no problem. And DHAP to glycerol 3-phosphate is a very simple reduction because here is a OH group, here is a ketone, right? Simple reduction. Additionally, if in your diet you got glycerol in your diet, okay, then you can directly utilize that glycerol as well. Okay, you can do a kinase reaction, use ATP, and then you would phosphorylate your C3. Okay, and then you get this glycerol 3-phosphate, right? And then glycerol 3-phosphate can then assimilate fatty acids. Now, before your fatty acids can react, what you need to do is first turn fatty acids into fatty acyl-CoA. Okay? Again, as always, organic acids are very stable, so they usually don't do much reaction. So if you want to react organic acids, you want to activate them into a thiol ester, typically coenzyme A. 可以哈，这个要记得哦，这还蛮，就是我讲很多次，所以应该会记得。You can add 1 arm to glycerol, 2 arm to glycerol, okay? And then you form this thing, okay? Okay? So the phosphate is still here, okay? When you add the acyl-CoAs, you're adding to this carbon, okay? The phosphate is still there. Now you get this thing called the phosphatic acid. 好难念。Right, but whatever 啦，就它基本上就是2个 arm，OK， 两个 lipid arm， 然后有一个 head group， 它上面是一个 phosphate，OK， 那这个我们考完试之后开始要讲那个 membrane lipids， 那这是一个很 basic 的 membrane lipid，right？ 它这边是2个 arm 嘛，然后这边是这样子，然后这边是一个 phosphate，right？ 所以这就是这个，所以它是最基本的 membrane lipid 的其中一个。Okay, so of course you can think about how we can use phosphatic acid directly for membrane lipid synthesis, or you can then take it. Okay, so this guy is a phosphate. You can take it, you hydrolyze it. Okay, you get rid of the phosphate into an OH, and then you can add another arm to it. Okay, and then you will get your TAG. Of course, this guy typically goes to membrane lipids. So that's it. Okay, that's all I have.